The clinical trial journey. A clinical trial investigates the safety and efficacy of a new medical intervention, such as a drug or a new medical device, in human subjects. The research generally consists of preclinical trials and 4 clinical phases, phase 1 to phase 4, in which a whole team of researchers, healthcare providers, research volunteers, and patients collaborate. The beginning of the journey starts with preclinical studies or discovery science, which happen before the clinical studies involving humans. Using in silico, in vitro, and in vivo methodologies, researchers aim to determine a starting safe dose for first-in-human study and to assess the potential toxicity of the product, but also to demonstrate proof of concept. On average, only 1 in 10 potential treatments from preclinical studies reach the clinical stages in humans. From there, only 1 in every 5,000 compounds that enter the drug discovery process will make it through the next phases and become an approved drug. There are instances where active substances that fail the clinical phase of their development due to their toxicity or insufficient efficacy go through the process of repurposing. This process involves finding a new target disease for the said substance, different from what the drug was originally designed and synthesized for. An example of such a substance is sildenafil, which was initially investigated as a potential antihypertensive drug. Sildenafil was shown to produce vasodilation and to inhibit platelet aggregation by inhibiting phosphodiesterase type 5, the enzyme that degrades cyclic GMP, and thus was considered a potential treatment for angina. However, during clinical trials, the drug caused an unexpected side effect in the form of penile erection. This physiological effect led to sildenafil being marketed as a treatment for erectile dysfunction under the brand name of Viagra. Subsequently, sildenafil was repurposed for a second time as a treatment for pulmonary arterial hypertension. The next phase is a phase 0 trial, which requires a small number of people, between 10 and 15 healthy volunteers. Also known as human microdosing studies or exploratory studies, phase 0 trials are designed to speed up the developmental process by verifying very early on if the effects of a promising new drug in human subjects are the ones that were expected from preclinical studies. Being by definition a dose too low to cause any therapeutic effect, a phase 0 study gives no data on safety or efficacy. Although welcomed by many for both scientific and animal welfare reasons, phase 0 trials are not considered a mandatory requirement for a clinical trial and are not widely used. Phase 1 trials, or first-in-human studies, require between 20 and 100 healthy volunteers, so the subjects will not see any benefit from the drug. Phase 1 clinical studies are primarily focused on questions regarding pharmacovigilance and mechanism. This phase assesses the safety, tolerability, pharmacokinetics, and pharmacodynamics of a drug. Because of the seriousness of high internal validity in matters of safety, Phase 1 studies require tight experimental control. After the drug is administered, the subjects are normally observed until several half-lives of the drug have passed, where half-life is the time it takes for the amount of that drug in the body to be reduced by exactly 1/2. Phase 1 trials usually involve dose ranging, also called dose escalation studies. The researchers start administering the drug at the lowest dose and escalate the dose levels until they discover the point at which the substance starts showing unacceptable side effects. The aim of this process is to establish the best and safest dose that can be administered in human subjects. The range of doses tested will typically be a fraction of the dose that caused a negative effect in animal testing. The dose must be at least 10 times lower than the no observable adverse effect level in preclinical studies, which is the maximum dose where there was not an observed adverse effect, and must be less than 10% of the predicted therapeutic dose. The extrapolation of doses in animals to humans is done through allometric scaling, an empirical approach that takes into consideration body weight, blood volume, half-life, and other pharmacokinetic data of human versus animal model. When translated to human subjects, the dose levels range depending on the animal used in preclinical studies. This stage can last several months to a year. Between 60 and 70% of the drugs from phase 1 studies go into phase 2, and only 10-15 become treatments. After a dose is established, researchers test in a phase 2 trial if the substance has any biological activity or effect on a larger group of human subjects, between 50 and 300 volunteers who have the target disease. The purpose of a phase 2 trial is to continue the safety assessments and to evaluate how well the drug works against the target disease. At this stage, the eligibility criteria becomes much more restrictive. It is usually during a phase 2 trial when the development process of a new drug fails, after researchers discover that the efficacy of the drug tested is not as good as it was predicted, or the drug has unacceptable side effects. Sometimes, in a phase 2 trial, a new treatment is compared with another treatment already in use, or with a placebo, thus making the trial a controlled one. Some phase 2 trials are also randomized, meaning that the researchers split the people taking part into treatment groups or cohorts at random. The best randomized trials are double-blind trials in which neither the volunteer nor the doctor knows in which treatment group the subject is in. A phase 2 clinical trial can last about 2 years. If the results from Phase 2 are encouraging, the journey continues to Phase 3 trials. These trials compare new treatments with the best currently available treatment, the standard treatment. The aim of a Phase 3 trial is to answer the following questions. Is the drug really safe? Does the drug really work in patients? And is it better than the current treatments? They involve many more patients than Phase 1 or 2 trials, from 300 to up to 3,000 participants that have the condition that the new medication is meant to treat. Depending on the targeted disease, a Phase 3 trial can last between 1 and 4 years. Because of the larger number of volunteers involved and longer duration, Phase 3 trials are the most expensive, time-consuming, and difficult trials to design and run. A study assessing the cost of more than 700 clinical trials run by 7 major pharmaceutical companies between 2010 and 2015 found that the average cost for a Phase 3 trial was $21.4 million, whereas the average cost for a Phase 2 and a Phase 1 trial were $8.6 and $3.4 million, respectively. Very commonly, while its regulatory submission and authorization for marketing are pending, Phase 3 trials will carry on, allowing patients to continue receiving a potentially life-saving treatment until the drug can be purchased. The last stage of a clinical trial is a Phase 4 trial, which involves thousands of participants. Phase 4 trials are conducted once a product has been approved for use and it is on the market, hence why it is also called post-marketing surveillance trial. This phase involves the safety surveillance, aiming to find out more about the side effects and safety of the drug, what the long-term risks and benefits are, and how the drug works when it is used more widely out in the real world, and allows rarer side effects to be assessed. An example of a drug that failed in the Phase 4 of a clinical trial is thalidomide, which was developed in the 1950s. Originally designed as a sedative, thalidomide was later marketed as a treatment for other conditions such as colds, nausea, and morning sickness in pregnant women. However, the drug was never tested in pregnant women, and as a result, researchers did not discover the teratogenic effects the drug had. Licensed for over-the-counter sales, thalidomide caused approximately 2,000 children deaths and birth defects in over 10,000 children worldwide. It took scientists 5 years to discover the link between thalidomide and the toxic effect it had on the embryo and fetus. Thalidomide was later repurposed as a treatment for a number of cancers and complications of leprosy. Phase 4 trials do not have a time limit. Doctors need to report side effects back as an ongoing process for all drugs. Some rare side effects can become apparent even after 10 years or more. Only 1 in every 5,000 compounds that enter a clinical trial becomes an approved drug and enters the market. The journey involving preclinical studies and the 4 phases of a clinical trial is a complex, time-consuming, and expensive one. Depending on the target disease, some clinical trials can cost up to $346 million and can last up to 12 years. However, as we have recently seen in the development of vaccines against COVID-19, the process can be sped up and last a year when the conventional route is fundamentally redesigned and nations collaborate. Nevertheless, it is not just a phase when discussing clinical trials. No less important, the ethical issues that arise in the development process of a new drug need to be reviewed.